Hi everybody, this is Miss Moriarty and I'm here to discuss with us our instructional video on some of the steps of meiosis and more vocabulary associated with this process. Now remember, right, meiosis we know is the process of creating gametes or sex cells. In the end, we're going to have 4 unique daughter cells that are each haploid in their number of chromosomes. Remember, we kind of saw this today in our modeling activity. We know that the chromosomes we find right in our cells are going to carry genes which control the physical traits of someone. It's chromosomes that we know come in matching sets, and we call those matching sets homologous pairs. And you can see that right in the karyotype or picture of someone's chromosomes here. We can see all of the pairs are matched up according to their size and shape, their banding patterns, and where their centromeres may be. Now, something really important to understand involving homologous chromosomes. We know that, especially in prophase 1, or in the very beginning of meiosis, those chromosomes are going to pair up. Now, the place in which they pair up is called the synapsis. They're going to consist of 4 chromatids in total, aka, which is why we call them tetrads. They are going to be visible during this stage of meiosis. One pair we know is going to be inherited from dad, while the other pair is inherited from mom, paternal and maternal. We know they are paired up because of their identical shape, size, and gene location, right? They may have the same genes, but they could potentially have different alleles. For example, homologous pairs of chromosomes can each carry the gene for eye color, but dad can have brown eyes and mom can have blue eyes. And we know during prophase 1, it's those genes that are going to cross over and swap or exchange DNA with one another, which helps to produce variety. So here are our homologous chromosomes. All right, so here's just another visual, right? We know that those homologous chromosomes exchange those genes. The point at which they do this is called the chiasma. So that is the point in which the crossing over actually occurs. And this is important because this is what provides us that genetic variation in the end, right? Those 4 unique haploid cells. Now, some other important vocab words for us to know are the 2 types of meiosis that we see in males versus females. In females, this is meiosis, known as oogenesis. This is the meiosis that's going to create eggs, or ova. Now, something slightly different between males versus females is that in oogenesis, only one cell will develop into an egg, while the other 3 are called polar bodies, and they will actually eventually die off. So we know, right, in meiosis, it always is going to produce 4 cells, but in oogenesis, only one of those egg cells is going to be viable, meaning it is going to be usable and will most likely be fertilized by a sperm cell. Now, in males, we call this spermatogenesis. This is the formation of sperm or meiosis in males. You'll notice here we have 4 sperm cells or 4, right, sex cells being produced, each being haploid, each having somewhat genetically different DNA. And eventually we know it's that sperm that's going to then fertilize that viable egg cell and eventually become a zygote. All right, guys, that's it for this video for today. You want to make sure that you have taken notes on your cell division notes and be ready to discuss these vocab terms further.